Hello everyone, this is Tejashri Dinakaran. My roll number is 7526. My topic for today is DNA methylation, a gene regulatory mechanism in eukaryotes. DNA methylation is a biological process by which methyl groups are added to the DNA molecule. Methylation can change the activity of a DNA segment without changing the sequence. When located in a gene promoter, DNA methylation typically acts to repress the gene transcription. In mammals, DNA methylation is essential for normal development and is associated with a number of key processes including genomic imprinting, X chromosome inactivation, repression of transposable elements, aging, and carcinogenesis. Two of the Two of the DNA's four bases, cytosine and adenine, can be methylated. Cytosine methylation is widespread in both eukaryotes and prokaryotes. Adenine methylation has been observed in bacterial, plant, and recently in mammalian DNA. Natural enzymatic DNA methylation takes place in adenine and cytosine. The modified bases are N6 methyladenine, 5 methylcytosine, and N4 methylcytosine. A methylation of cytosine to form 5-methylcytosine occurs at the same fifth position on the pyrimidine ring where the DNA base thymines methyl group is located. The same position distinguishes thymine from the analogous RNA base uracil, which has no methyl group. Spontaneous deamination of the 5-methylcytosine converts it to thymine. This results in a TG mismatch. Repair mechanism then correct it back to the original CG pair. Alternatively, they may substitute adenine for guanine, turning the original CG pair into a TA pair, effectively changing a base and introducing a mutation. Before going to the mechanism of DNA methylation, we need to first know the term CPG islands. CPG islands are regions with higher concentration of cytosine and guanine base pairs. Enzymes that carry out methylation are called as DNMT or DNA methyltransferases. DNMT1 maintains methylation while DNMT3A and 3B carries out de novo methylation. DNA methylation may affect the transcription of genes in two ways. First, the methylation of DNA itself may physically impede the binding of transcriptional proteins to the gene, and second, the likely more important methylated DNA may be bound by proteins known as methyl CpG binding domain proteins, which are also known as MBDs. MBD protein then recruit additional proteins to the locus such as histone deacetylases and other chromatin remodeling proteins that can modify histones, thereby forming compact inactive chromatin termed as heterochromatin. This link between DNA methylation and chromatin structure is very important. This slide shows a general pathway of how the methyl group has been transferred to cytosine from SAM to SF via SAH. DNA methylation lies at the heart of a phenomenon called as imprinting. In a diploid cell, there are two copies of most genes. One copy on a chromosome inherited from mother and other inherited from father. In most of the cases, the two alleles are expressed at comparable levels. They carry the same regulatory sequences and are in the presence of same regulators. They are also located in an equivalent region of two very similar chromosomes, but there are a few cases where one copy of a gene is expressed while the other is silent. Two well studied examples are the human H19 and IGF2 genes, which is shown in the figure. These are located close to each other on the human chromosome number 11. In a given cell, one copy of H19 that is on the maternal chromosome is expressed while the other copy on the paternal chromosome is switched off. For IGF2, the reverse is true. The paternal copy is on and the maternal copy is off. Two regulatory sequences are critical for the differential expression of these genes: an enhancer, which is downstream of the H19 gene, and an insulator located between the H19 and IGF2 genes. The enhancer, when bound by the activators, can activate either of the two genes. So, why does it activate only H19 on the maternal chromosome and IGF2 on the paternal chromosome? The answer lies in the role of the insulator and its methylation state. The enhancer cannot activate the IGF2 gene on the maternal chromosome because on that chromosome the insulator binds a protein known as CTCF that blocks the activators at the enhancer from activating the IGF2 gene. On the paternal chromosome, in contrast, the insulator element and the H19 promoter are methylated. In that state, the transcription machinery cannot bind the H19 transcription cannot bind the H19 promoter, and CTCF cannot bind the insulator. As a result, the enhancer now activates the IGF2 gene. The H19 gene is further repressed on the paternal chromosome by the binding of a protein known as MECP2 to the methylated insulator by recruiting deacetylases, and these repress the H19 promoter. Disease conditions. First one is cancer. So there can be some abnormalities in DNA methylation which can lead to disease conditions like cancer and atherosclerosis. So first one is cancer. It is commonly known that inactivation of certain tumor suppressor genes occurs as a consequence of hypermethylation within the promoter regions, and numerous studies have demonstrated a broad range of genes silenced by DNA methylation in different cancer types. Altered expression of microRNAs also silence or activate many genes in progression to cancer. Altered microRNA expression occurs through hyper or hypomethylation of CpG sites in CpG islands in promoters controlling transcription of the microRNAs. Next is atherosclerosis. Two of the cell types targeted for DNA methylation polymorphisms are monocytes and lymphocytes, which experience an overall hypomethylation. One proposed mechanism behind this hypomethylation is elevated homocysteine levels, which can cause hyperhomocystemia, a known risk factor for cardiovascular disease. High plasma levels of homocysteine inhibits DNA methyltransferases, which causes hypomethylation. Hypomethylation of DNA affects genes that alter smooth muscle cell proliferation, causes endothelial cell dysfunction, and increases the inflammatory mediators, all of which are critical in forming atherosclerotic lesions. The last slide here shows the references I referred to while making this PPT. Thank you.